The tent still smells like kerosene and wet clay, decades after anyone struck it. Somewhere in a storage vault beneath the British Museum sits an archive box marked simply Ur, one of thousands pulled from the ground in the 1920s under the direction of Leonard Woolley. And inside that box, and 100 others like it, sits a question nobody in a lab coat has ever been able to close. Where did the Sumerians actually come from? For a century, that question has haunted Mesopotamian scholarship because the language itself refuses to help. Sumerian is a linguistic isolate, unrelated to anything else spoken on Earth, ancient or modern, and that isolation opened the door to a parade of theories. Traders arriving from across the Indian Ocean, migrants pressing in from Central Asia, fringe claims, at their most extreme, of a people arriving from somewhere entirely outside the region, no traceable homeland at all. All of this unfolding on top of one particular stretch of land, the delta carved out by the Tigris and the Euphrates before they empty into the Gulf, ground so associated with fertility and origin that biblical tradition named it the Garden of Eden. For most of the 20th century, none of the theories about who built the first cities there could be tested against anything harder than pottery style and grammar. Skeletons existed, genomes did not. That only changed technically in roughly the last decade, when sequencing finally advanced far enough to pull usable DNA out of bone that old, buried in soil that unforgiving. So after 100 years of educated guessing, dig permits, and archive boxes with no answer inside them, the question finally had a way to get answered directly. What does the actual genome say? The lab results started coming in, and they didn't confirm any of the old guesses. They rewrote the question entirely. If you're the kind of person who'd rather trust a genome pulled out of a 2,000-year-old tooth than a 100-year-old guess written in a museum ledger, this is exactly the kind of place worth sticking around for. Hit subscribe, because the digs keep coming, the labs keep publishing, and this particular mystery has a lot more layers still buried under it. This isn't a single reveal and done. It's a whole map of sites, spread across centuries, each one adding a piece that the last one couldn't see. Some of what's coming will confirm what archaeologists already suspected from pottery and inscriptions. Some of it will contradict a theory that's been repeated so often it started to sound like fact. That contrast, evidence against assumption, is the entire spine of what follows here, and it plays out one skeleton, one river valley, one archive at a time. So settle in. There's dirt still left to turn. Now, back to the dirt. The expectation, going in, was simple enough to fit on an index card. One cradle, one founding population, one clean genetic line running back to the first farmers who settled the delta. A single stock, traceable, tidy. The genomes recovered from Çayönü broke that expectation almost immediately. Çayönü sits in what's now Diyarbakir, in southeastern Turkey, and researchers working under the EU-funded NeoGene project, led on the anthropology side by Associate Professor Ezgi Altınışık of Hacettepe University, pulled genetic material from skeletons dating back to 8,500 BCE. What they found wasn't a single population slowly filling in. It was diversity that was already high at that early date, and diversity that stayed remarkably stable across roughly 1,000 years afterward, rather than collapsing into one dominant lineage the way a simple founder story would predict. One burial in particular stood out. A 2-year-old girl whose genetic makeup traced closer to communities on the eastern side of the Fertile Crescent rather than the population she was buried among. That doesn't happen by accident. It means her family, or the generations before her, had moved. Çayönü sits only about 300 kilometers from Göbekli Tepe, placing it in the same broad Neolithic world, and yet the genetic profile at each site reads as distinct. The clearest signal out of all of it is directional. The data shows a flow of ancestry moving from Mesopotamia into the interior of Anatolia, not the reverse. Move roughly 400 kilometers east, into the mountains, and the picture splits apart completely. Ganj Dareh sits in the Zagros Mountains of Iran, and in 2016, Brushaki and colleagues published 4 genomes from that site in the journal Science, dated to roughly 10,000 years before present. These weren't a regional variant of the same founding population seen elsewhere. The Zagros farmers came back genetically distinct from both the Anatolian and Levantine farmers living at the same time, and just as tellingly, they made no meaningful contribution to the ancestry of later Europeans. That's a genetic dead end running in one direction while everything continues elsewhere. It doesn't mean the population disappeared. It means their descendants went a different way, north into the Eurasian steppe rather than west into Europe. Archaeologists had generally assumed something closer to cultural diffusion, An idea and a set of farming techniques spreading outward from a single point of origin. The genomes argue for something else entirely. Not one migration, but several genetically separate populations independently taking up farming across the same broad stretch of territory, at close to the same time. Not diffusion, population structure. 3 separate founding populations, all sitting under one geographic label, and that's before Mesopotamia has built a single city. The core finding sitting underneath every one of these papers is this: Mesopotamia was never one gene pool. It functioned as a genetic crossroads, sitting at the connective point between Anatolia, the Levant, the Caucasus, and Iran, and every population passing through left a trace. The data draws a real boundary too, a documented east-west genetic structure running between Upper Mesopotamia and the central Zagros, marked by the lowest migration rate measured anywhere in the entire dataset. 2 populations living in relatively close proximity, largely not mixing. And yet the Çayönü child shows that boundary wasn't absolute. Individual movement across it happened, on a personal and family level, even while the broader populations stayed genetically apart. Diversity and structure existed at the same time, high genetic variation across the region as a whole, paired with real, persistent barriers in specific pockets. That's the secret sitting inside this title. Not one hidden ancestor waiting to be found, but a fault line pattern of exactly who mixed with whom and who didn't. Ur-3 period administrative texts already described multiple ethno-linguistic groups living side by side across southern Mesopotamia: Sumerian, Akkadian, Hurrian, Amorite, all documented in cuneiform archives long before anyone could test it genetically. Now the DNA gives that written record an actual biological backbone. The 2016 genomes named one more player in this story, and it changes everything east of Mesopotamia. In 2015, Jones and colleagues published genomes recovered from western Georgia that gave this story its next name. One individual, nicknamed Satsarblia, dates to the late Upper Paleolithic, roughly 13,300 years old. Another, nicknamed Codius, comes from the Mesolithic, roughly 9,700 years old. Together, these 2 skeletons define what researchers now call the Caucasus hunter-gatherer lineage, or CHG. A distinct ancestral branch that simply didn't exist in the scientific record before these genomes were sequenced. CHG ancestry doesn't stay confined to the Caucasus. It sits at the root of the Neolithic Iranian farmer genomes discussed a moment ago, the same Zagros population that split off as its own genetically distinct group. Modeling of these lineages shows CHG carrying roughly 10% more ancient North Eurasian-related signal than the Neolithic Iranians who trace back to a related source population. And CHG doesn't stop there either. It becomes one of 2 core ingredients, alongside Eastern hunter-gatherer ancestry, that together form what's known as steppe ancestry, the genetic signature that would eventually make its way back down into Mesopotamia during the Bronze Age. No skeleton recovered from Sumer itself has ever tested as purely Caucasus hunter-gatherer. That lineage never arrives whole. It arrives folded into other populations, diluted, mixed, carried forward indirectly into whoever eventually settled the delta. A hunter-gatherer from a cave in Georgia, living centuries before Sumer existed as an idea, is quietly present in the ancestry of the people who built it. The genomes recovered from Çayönü and the Zagros solved one problem and immediately opened another. Near Eastern populations simply didn't fit cleanly onto the family tree connecting European and East Asian hunter-gatherer lineages, and something was missing from the math. Lazaridis and his colleagues worked backward from the patterns embedded in these Near Eastern genomes, and mathematically inferred an entire population to account for the gap, a lineage now called Basal Eurasians. Their ancestry, by some models, accounts for as much as 38 to 48% of certain Neolithic Iranian-related genomes, a substantial share of the genetic makeup running underneath everything discussed so far. And despite that level of statistical certainty, despite entire population models built around their existence, not one Basal Eurasian skeleton has ever actually been recovered. The population that helped shape the ancestry of the Cradle of Civilization is, physically, still missing. Known only through its genetic shadow, detectable in everyone who came after, never held in anyone's hands. This ghost lineage sits underneath both the Zagros and Iran branch already covered, and more distantly, underneath the Levantine branch as well, a shared root running beneath populations that on the surface look genetically separate from one another entirely. With that shadow population accounted for on paper, the next real advance needed actual bones, and Armenia is where researchers finally got them. Iosif Lazaridis and his team at Harvard University recovered genome-wide data from Neolithic individuals near two sites in Armenia, Basal Eurasians and Akhnashan, dated to roughly 6,000 to 5,000 BCE. What made this find valuable wasn't just its age. It captured a moment right before regional admixture erased the differences between populations. A genetic snapshot researchers describe as unusually well-timed for exactly that reason. One individual, buried at Akhnashan, carried ancestry dominated by the local population. A second, buried at Basal Eurasians, carried ancestry linked instead to Mesopotamia proper, despite living in the same valley during the same era. The broader study also traced two separate migration pulses, moving from the Fertile Crescent heartland into Anatolia. An earlier pre-pottery pulse drawing from Mesopotamian-related and local sources, followed later by a pottery-era pulse that added Levantine-related ancestry into the mix. The same research suggested something else worth noting. Early farmers on Cyprus trace back to Anatolia rather than the Levant, a single detail that reroutes an entire migration path archaeologists had assumed ran differently. Two neighbors, two different ancestral stories, living in one valley. That kind of contrast stops being the exception and becomes the rule once cities start to appear. A 2026 study published in Genome Biology, led by Matthew P. Williams, Peter A. Miglis, Alan J. Cooper, and their co-authors, sequenced 17 individuals from Bakr Awa, a site in northeastern Iraq spanning both the Bronze and Iron Ages. Bakr Awa sat directly on the frontier between Mesopotamia and Iran, and its cuneiform archive preserves texts written in both Akkadian and Hurrian, physical proof of linguistic mixing that the DNA now matches step for step. The Bronze Age genomes from this site show a marked shift: Anatolian ancestry, Levantine ancestry, and Caucasus and Yamnaya steppe-related ancestry, all layered onto the older local population. That heterogeneity is a sharp break from the site's earlier pre-pottery Neolithic genetic profile, which had closely matched nearby Best Answer and the Zagros-related ancestry already covered. The site's architecture shifted right alongside its genetics. Local tradition gave way to Akkadian period design around 2350 to 2150 BCE, then shifted again toward renewed southern Mesopotamian influence once the Neo-Sumerian Ur-3 kingdom rose to power. Cultural change the genomes now track almost step for step. A devastating fire brought Bakr Awa's Bronze Age occupation to an end sometime in the 15th or 14th century BCE, and the population that followed in the Iron Age shows that earlier Bronze Age diversity being integrated rather than replaced. Steppe ancestry reaching this far into northeastern Iraq means the story now has to follow the Yamnaya Trail north to find where it started. Steppe ancestry itself forms from a mixture of Eastern hunter-gatherer ancestry and CHG, or Iranian-related ancestry, first documented in Neolithic individuals from the Samara region, dated between 5,200 and 4,000 BCE. That exact steppe signature is the same ancestry component detected moments ago in the Bronze Age Bakr Awa genomes, meaning the steppe's genetic reach extended as far south as northern Iraq. The mechanism researchers propose isn't a single mass migration sweeping through. It's a layered sequence, Caucasus populations arriving first, steppe-descended groups following, both mixing repeatedly with the older Fertile Crescent base rather than replacing it outright. Genetically, this places Mesopotamia at the southern terminus of a corridor running from the Eurasian steppe, down through the Caucasus, into the Tigris-Euphrates basin itself. Historically, that corridor lines up with incursions and trade contacts already well documented outside the genetic record. Hurrians, Mitanni chariot elites, and later Indo-Iranian-speaking groups, all moving through this same stretch of geography over time. Not one Bronze Age Mesopotamian genome tested so far comes back purely steppe-derived. Everyone is a blend, consistent with ongoing contact stretched across generations, rather than a single conquest followed by replacement. Contact ran the other direction too, south, into a river valley where nobody expected to find Mesopotamian blood waiting in the ground. In July 2025, Nature published something that had never existed before in the history of Egyptology: a complete ancient Egyptian genome. Linus Girdland Flink of the University of Aberdeen and his colleagues sequenced it from a man buried at Nuweirat, radiocarbon dated to between 2055 and 2570 BCE. The burial itself mattered as much as the science. He'd been interred inside a ceramic pot, placed within a rock-cut tomb, a combination that created just stable enough conditions for his DNA to survive across roughly 4,500 years. What the genome revealed was a clean split, Approximately 80% of his ancestry traced to ancient North Africa, and approximately 20% traced to the eastern Fertile Crescent, specifically Mesopotamia. Before this single genome, only 3 partial ancient Egyptian genomes existed anywhere in the scientific record, and all 3 came from a much later window, 787 BCE to 23 CE, over 2,000 years after this man lived. Isotope analysis of his tooth enamel filled in the rest of the picture. Wheat, barley, legumes, animal protein, a diet consistent with the Nile Valley. He wasn't passing through, he lived his life there. The genome even let researchers infer his physical traits: brown eyes, brown hair, dark skin. Decades earlier, in the 1980s, Svante Pääbo attempted to extract DNA from Egyptian mummies and failed outright, years before he'd go on to win a Nobel Prize for sequencing the Neanderthal genome. It took another 40 years of advances in sequencing technology to finally get a result Pääbo couldn't reach. 1 skeleton, 1 tooth, and roughly 100 years of archaeological guesswork about contact between Egypt and Mesopotamia just got its first hard genetic confirmation. Archaeologists have argued for well over a century that Egypt and Mesopotamia were in contact, pointing to shared artistic motifs, imported cylinder seals, and architectural parallels that show up in early dynastic Egyptian material culture. All of that was circumstantial, built from objects and style. The rewrote genome is the first time that proposed contact comes with genetic confirmation attached to it, rather than resting on artifact comparison alone. His Mesopotamian-related 20% places him squarely within the time frame of the Old Kingdom, bridging the early dynastic period and the era when the Great Pyramid of Giza was under construction. An ancestry pattern like that doesn't come from trade goods changing hands. It implies actual gene flow, meaning people crossed that distance, not just objects. At the same time, most of his genome is best represented by North African Neolithic ancestry among the reference genomes currently available. So the local population clearly remained dominant even while absorbing this outside contact. Researchers were careful to point out how fragile this find really is. The tomb's preservation was close to accidental, a reminder that most of the ancient DNA from this era simply hasn't survived at all, which means what's been recovered so far may only be the surface of a much larger pattern. If contact reached that far west, into the Nile Valley itself, it's worth asking what reached the cradle from the opposite direction, deep into South Asia. A study published in PMC examined mitochondrial DNA recovered from 4 individuals excavated at Tel Ashara, the site of ancient Turka, and Tel Masaik, ancient Kar Asurna Sirpal, both in the middle Euphrates Valley of Syria. The remains span an enormous stretch of time, roughly 2,500 BCE through 500 CE, running from the early Bronze Age all the way into the late Roman period. The genetic material recovered carried mitochondrial haplogroups M4B1, M49, and M61, lineages that originally arose on the Indian subcontinent during the Upper Paleolithic. Those same haplogroups are completely absent from Syrian populations living today. Whatever thread connected these individuals to South Asia was cut at some point in the centuries since. And yet the haplogroups themselves didn't vanish entirely. They persist today in Tibet, the Himalayas, India, and Pakistan, meaning the genetic signal survived, just relocated thousands of kilometers from where it was actually found in the ground. Researchers proposed a few possible explanations. Descendants of very old migration waves out of the subcontinent, or long-distance merchants who moved along trade routes running through the region and simply never left. Older, now largely superseded theories have occasionally lay the whole timeline out in order, and a rhythm starts to show through the noise. In the Neolithic phase, roughly 10,000 to 6,000 BCE, the founding populations were genuinely separate. Zagros, Levant, Anatolia, each one largely descended from its own local hunter-gatherers, with minimal cross-mixing between them. Then comes the Chalcolithic phase, and with it, the first major homogenization. Chalcolithic Western Iran forms from a documented 3-way blend: local Neolithic ancestry, Levantine ancestry, and Caucasus hunter-gatherer ancestry, all combining in one population for the first time. That Chalcolithic Iranian-related ancestry doesn't stay put either. It radiates outward, contributing an estimated 44% of the ancestry found in Levantine Bronze Age populations, and roughly 33% of the ancestry in Chalcolithic Northwest Anatolians. By the Bronze Age, the pattern grows more complex still. Steppe and Yamnaya-related ancestry arrives from the north, visible directly in the Bakr Awa genomes, while contact with Egypt through the rewrote individual, and possibly with South Asia through the Turka, remains extends outward in entirely different directions, all happening at roughly the same time. Then the Iron Age arrives, and the Bakr Awa genomes show something different again. The heterogeneity built up across the Bronze Age becomes integrated into a more unified population following resettlement, diversity converging instead of staying fragmented. And in the present day, the Marsh Arabs preserve a strong autochthonous signal, evidence that beneath every wave of contact recorded here, a core local lineage persisted continuously, from the Neolithic all the way through to the people living in southern Iraq today. That's 5,000 years of contact events layered on top of one another, which is a lot of history for a single skeleton to carry. But the youngest resident of Çayönü carried exactly that. Return to her for a moment. The 2-year-old girl buried at Çayönü, dated to roughly 8,500 BCE, whose genome showed ancestry from the eastern side of the Fertile Crescent, despite the fact that she was buried in the north. Her genome is, in miniature, the entire pattern this investigation has been tracing from the start. A local population, cross-regional ancestry folded into it, and movement happening a full 6,000 years before anyone in this region had written a word down. The same dataset that identified her ancestry also let researchers examine a young girl's skull from that same broader Çayönü collection and identify evidence of primitive surgical practice, specifically cauterization, meaning the genetic record and the medical record overlap directly at this one site. That combination, deep genetic mixing sitting alongside evidence of medical practice this early, reframes Çayönü. It stops looking like a simple early farming village and starts looking like a genuinely cosmopolitan settlement by Neolithic standards. And that lowest migration rate detected anywhere in the region, the one sitting between Upper Mesopotamia and the central Zagros, still didn't stop individual movement from happening across exactly that boundary, case by case, family by family. Structure and contact were never opposing forces here. They operated at the same time, just at different scales. Every named site in this investigation, Çayönü, Ganj Dareh, Masisblur, Bakr Awa, Nuweirat, Terka, the marshes of southern Iraq, sits on one connected map, and not one of them tells quite the same genetic story as the next. Plenty here still doesn't have an answer. No individual who genetically tests as Sumerian has ever been found buried alongside a Sumerian language cuneiform text in the same grave. So the genetic record and the linguistic record haven't yet been matched to one single named person. The Basal Eurasian population remains entirely unrecovered as physical remains, inferred through mathematics, never actually held in anyone's hands. The South Asian-linked mtDNA haplogroups found at Turka raise a question no current study has settled, whether those individuals were migrants, traders, or descendants of a much older population nobody has otherwise sampled at all. Sample sizes across this entire field remain small, 4 individuals at one site, 17 at another, meaning a single new genome still has the power to shift the whole picture again. Most of the ancient DNA recovered from Egypt and Mesopotamia alike depends on unusual preservation conditions, the same kind of sealed pot burial that saved the Nuweirat genome, meaning what survives to be studied is a matter of accident rather than design. Excavations continue across northern Iraq and the wider Fertile Crescent, including further work tied to the Neogene project, and researchers expect more genomes to surface in the years ahead. The century-old Sumerian problem didn't get a final answer here. It got its first real data. The myth this investigation opened with, a single mysterious founding population behind the Cradle of Civilization, doesn't survive contact with the genomic record. The reality that replaces it is more complicated and more interesting. Multiple genetically distinct populations occupying adjacent territory, real boundaries standing between some of them, real contact and admixture running across others, all of it layered continuously across roughly 7,000 years. The Marsh Arabs carry a real, datable genetic echo of the Sumerian period itself, tied to a specific Y chromosome branch that lines up with the timing of the city-states. The Nuweirat genome proves that Mesopotamian contact reached the Nile by the Old Kingdom. The Zagros farmers prove that the Neolithic transition was never one migration, but several, unfolding in parallel across the same broad stretch of territory. The child buried at Çayönü proves that individual movement was already happening 6,000 years before any of these civilizations even had a name. None of that was written in the pottery. All of it was written in the teeth. Whole genome sequencing is now capable of recovering complete Egyptian and Mesopotamian genomes from a single tooth. And the next decade of excavation is likely to add names, sites, and individuals to this map far faster than the last 100 years managed. Ongoing work at sites like Bakr Awa suggests northern Iraq alone could still yield genomes spanning the entire Bronze to Iron Age transition in far greater detail than what's been recovered so far. The unresolved mtDNA connection found at Turka remains open, waiting for a future study to confirm it as long-distance migration or trace it back to some entirely different explanation. And the physical remains of the Basal Eurasian population, if they're ever actually found, would close one of the oldest open questions in this entire field. If a single tooth from one skeleton, sealed inside a pot for 4,500 years, could rewrite the relationship between Egypt and Mesopotamia overnight, it's worth wondering what else is still buried right now that would rewrite the rest of it. The Cradle of Civilization was never one bloodline, it was a crossroads, and the DNA finally proved it.